Hi everyone, let us study about the common and the famous protozoan disease, malaria. Malaria is a mosquito-borne disease caused by Plasmodium. It is transmitted by the bite of female Anopheles mosquito. Now, malaria is worldwide. Annually, hundreds of millions of people get infected, and it is reported that in 20th century around 700 million people were reported with malaria, out of which 3 million were died. These cases were very common in the regions of Asia and Africa, particularly in the India. Malaria is caused by species of Plasmodium, namely Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, Plasmodium malariae, out of which Plasmodium vivax is the most common cause of malaria. Now, the life cycle. It basically occurs in two different hosts. One is human and one is female Anopheles mosquito. Primarily, human host is infected by plasmodial sporozoites, which are small, elongated cells. These travel through the bloodstream and reach the liver. The stage is known as the schizoid, and these schizoids segment into number of cells called as merozoites. Some of these merozoites infect the red blood cells. During this period, symptoms such as chills, fever occur. Now, the protozoite cells that cannot infect the RBCs are called as the gametes. These gametes mature within the mosquito, and these gametes fuse to form a zygote. These zygote are then released, and some of them reaches the salivary gland of the mosquito, where these are being again deposited back to the humans, and the cycle continues. Now, pathogenesis. Clinical symptoms usually appear after 10 to 15 days of infection, which is characterized by chills, high fever, etc. The benign malaria caused by Plasmodium vivax is characterized by periodic illness interspersed with period of well-being, while the malignant malaria caused by Plasmodium falciparum exhibits the symptoms such as fever that is more persistent. Also, it results in edema of brain and blockage of kidney. The high fatality rate of malignant malaria is due to high rate of reproduction of the merozoites. Now, lab diagnosis of the Plasmodium infected RBCs requires the identification of the infected RBCs in blood smears of patients. Giemsa staining is used to recognize the parasite present in the infected RBCs under the microscope. Apart from these, nucleic acid probes, PCR methods, antigen detection methods are also some of the serological methods that are used. Now, what are the control measures? The obligate host, which is nothing but the female Anopheles mosquito, can be controlled by the insecticide, followed by treatment of patients with a drug, which destroys the Plasmodium life cycle. Also, it can be controlled by netting around the sleeping area. Mosquito repellents are also used, wherein the mosquito is being kept away from infecting the humans. Now, the treatment. Chloroquine and primaquine drug combination is effective in cure of malaria, wherein chloroquine destroys the merozoid within the RBCs and primaquine destroys both the sporozoids and merozoids in the liver and gametes outside the RBCs. If the combination of these drugs doesn't work, then Malaron is recommended. Also, antiparasitic compounds such as artemisinin is also used. Hope you guys understood the topic, the protozoal disease malaria. If you haven't subscribed to our channel yet, please do subscribe, like the video, share. Thank you so much for watching.